Hey guys, welcome back to Biology 1 Lab. So today what we have to do is finish up this lab on taxonomy, and I believe this is our last one. So much like the last time, I'm just going to read through it. I have some extra materials for you as well, and I will be using those materials to build it for you a quiz to take that will help you to study for your test. So you can be expecting all of those things by the time you see this video up. Alright, so let's begin. This is a lab on taxonomy, and the idea with taxonomy, let's go ahead and lay this all out there for you, is that in the past, what we have done is we have set up taxonomic keys, is what these are called, keys, so that we can have an organism run through what's referred to as a taxonomic key and identify that organism all the way down to species level. So kingdom, phylum, class, order, all the way down. All the way down to species. And it works pretty well, but once the, we'll call it genetic revolution hit, where we are capable of taking a tissue sample from an organism and running the genetics for that organism, it's different now. So rather than just using taxonomy, which is looking at, well, frankly, how organisms look, what we try to do now is to classify species based off of their genetic similarities to one another, not just the way they look. Because the way things look, man, I'm here to tell you, it's misleading sometimes. You get into situations that are referred to as, let's see, divergent and convergent evolution, for instance, and you end up with organisms that can look very similar to one another because they live in similar environmental conditions, but they are genetically dissimilar. So it's misleading. So taxonomy is useful today, but it is not the end use for identifying organisms by modern standards. Modern standards would be based off of genetic testing. Okay, so let's talk about taxonomy. Okay. In simple terms, taxonomy is defined as the science of naming and classification. So this is broken down into the field of systematics. Close related taxonomies overlap the field of systematics, which is a study of biodiversity. Asks, why are there so many species living on the earth and how did they develop? Alright, so in essence, what we're going to be talking about today is commonly referred to as the Linnaean system of taxonomy, or Linnaean taxonomy. It's from a long, long time ago. Back in the 1700s, this was initially devised. And Linnaean taxonomy is running from, well, actually, Linnaeus didn't go all the way up to domain. Today, in Linnaean taxonomy, we start with domains. So we have our domains, bacteria and eukarya. And then in domain eukarya, we have kingdom protista, fungi, plantae, and where's Animalia? Oh, it's a whole different subset. And Animalia. Alright, so key take-home messages. Domains, bacteria, archaea, and eukarya. Bacteria and archaea, these are prokaryotic. You need to understand this means that they do not have membrane-bound nuclei. Okay, prokaryotic organisms do not have membrane-bound nuclei. This is a key take-home concept of Biology 1. Domain eukarya, on the other hand, these would be organisms that are eukaryotic. Eukarya. That means true nucleus. True nucleus. Okay, so these have what we call membrane-bound organelles, like nuclei, for instance. And in domain eukarya, we have kingdom protista, kingdom fungi, kingdom plantae, and kingdom animalia. Now, protista is, as they say here, a grab bag. There are lots of different protists, as you well know at this stage. Like we have talked about kingdom protista before in one of the earlier labs. You've even seen a lot of protozoans, for instance, in your lab where we looked at pond water. You saw all those critters swimming around. So you've seen a lot of protists, and I'm here to tell you, you've only seen a subset of protists. They are very diverse. Kingdom fungi is pretty obvious. These are funguses like mushrooms, anything that grows in hyphae, as we described previously. Again, you need to be friends with this as a concept. Kingdom plantae would be the plants, green plants for the most part is what we're thinking about here. Everything from oak trees to the grass in the fields. Right? And then, of course, there's Kingdom Animalia. Now, Kingdom Animalia is a little different, and they go through and they describe a bit here. What I want to point out to you is that if I were you, I would know the classification scheme here. Phylum, class, order, genus, species, family, genus, species, I should say. And in this, I would understand in the main point of this lab, the concept of binomial nomenclature. Okay, binomial nomenclature. Let me tell you, I said on your first, I think, lab test, that I was going to make you give me a genus and species name, like what is a species name for an organism. If you didn't write it right, and if you didn't capitalize or do things in the correct manner, then I was going to count it wrong. And I ended up doing that for a lot of you guys, alright? I'm telling you again now, if you can't tell me how to write a genus and species correctly, like if I say, what's your species name? If you can't tell me how to write Homo sapiens correctly, we got major problems. Okay, so let's go through this. An organism's scientific name consists of two parts. The first part is the genus name, okay, genus, and then the second part is a species name, wherever that is in there someplace. Okay, that is binomial, two. Binomial means two. Two-part nomenclature. So, for instance, Felis leo, Felis leo. Felis would be a genus name, leo would be a species name. Quercus alba, all right, Quercus is a genus name, alba is a species name. And what I want you to notice about this is it's either going to be underlined or italicized. Underlined or italicized. Normally what you see is if someone is handwriting something, they underline it because it's kind of hard to tell italics versus non-italics with your handwriting. Whereas in a printed format of this fashion where it's all typed in, it's going to be italicized. You always italicize or underline species names. Okay, always italicize or underline. And further, you capitalize the genus and you lowercase the species. So Felis is capitalized, Leo is lowercased. Quercus is capitalized, Alba is lowercased. Okay, this is very important. And now, worthy of mention here is the concept of the common name. Common names are what people in a given area call a species, or a group of species in a lot of cases. The classic example of this is what we'd call water moccasins. So water moccasins have a genus and species name, but if you say water moccasin, people know what you're talking about. Kind of like if I say like an African lion. If I tell you, hey, think about an African lion, you'll think about, you know, the big mane and all that fun stuff. Or if I say Panthera leo, you won't really know exactly what I'm talking about. So common names are useful because they're the names that people utilize to talk about these organisms in most cases, but they can also be very misleading. So I've heard just a plethora of different common names for the same organism. People just make them up off the top of their heads like you would not believe. Yeah, I thought they had an example or two of that. But trust me when I say common names are not used in science. They're not used in science. All right, common names are not used in science because they can vary from location to location to location. They can vary from person to person to person. I'm here to tell you, I've heard a bunch of different common names for things that it just makes no sense. So, will I ask you for common names? Yes, I will. Just generally speaking. But my main goal is that you can give me genus and species. And for these genus and species, you know that the first one is capitalized, the second one is lowercase. Alright. Now, how do I identify these types of organisms? Using what we call dichotomous keys. Okay? Dichotomous. Dichotomous keys. We use dichotomous keys for the identification of organisms in most cases, especially when you're working in a field situation, like if you're out in a stream and you're trying to identify an organism on the spot, you're not running genetics out there, you have your dichotomous key you can run through to identify those critters. Now, they've given you a nice example of a dichotomous key here, and this is like how to identify something that you see in the world around you. Alright, so hang on, give me a second. And we're back. So what I've done is I've pulled up an image of a writing device. Let's pretend that we have no idea what this thing is, and we can use a dichotomous key to be able to figure it out. Now observe. A dichotomous key is always two parts. So there's 1A and 1B, 2A and 2B, 3A and 3B. What you do is you start at the beginning and work your way towards wherever you need to go. So, 1A, vehicle self-propelled with some type of engine or motor. Is this vehicle self-propelled with an engine or motor? No. So you go to 1B. 1B, vehicle propelled by muscular effort of rider, pedaling. Yes, that's correct. So then you go to number 3. All right, you go to number 3. So we skip the 2s, we go to 3. So if we go to number 3, part A, vehicle with only one wheel. Does this have one wheel? No, it doesn't. So we go to 3B, vehicle with 2 or more wheels. Yes, that's correct. 1, 2, 3 wheels. Vehicle with 2 or more wheels, we go to number 4. So we go to 4. 4A, vehicle with only 2 wheels? No, that's not correct. But if it only had 2 wheels, that would make it a bicycle. Vehicle with 3 wheels, 1, 2, 3, then this has to be a tricycle. That's how you run a dichotomous key. If this was a motorcycle, we'd say vehicle self-propelled with a motor. Yes, go to 2. Vehicle with only 2 wheels. Yes, then it's a motorcycle. If it had 4 wheels, vehicle with 4 wheels, it'd be an automobile, like a car. Okay, that is how we run dichotomous keys. And frankly, folks, that's really as much as you need to know out of this. Let me double check my notes here. So be able to ID organisms to phylum and class, which we'll be talking about here in just a second. Be able to tell me their common names. Be able to write a species name correctly. Yeah, be able to tell me the difference between prokaryotes and eukaryotes. Be able to tell me about the kingdoms in eukarya. Yeah, all right, perfect. So what I have for you is a brief breakdown of a pretty intensive dichotomous key that we will use in lab. It's got some terms here that we need to talk about. It goes through and identifies your procedure on how to do it. It has this chart. If you don't have a lab manual handy, this chart is going to be important for you. Okay? Phylum, class, and these are common names. Today, what you're going to be identifying on your lab quiz is a squid, a clam, a chitin, a marine clamworm, a freshwater leech, a sea cucumber, a sea urchin, an earthworm, a lubber grasshopper, a marine snail, a crayfish, and a sea star. Now, I don't have them necessarily in that order for obvious reasons, but what you'll have to do is tell me phylum and class for all of these. Okay, phylum and class for all of these. Now, how do you get your phylum and class? Well, you have to do a dichotomous key, and I'm going to provide this key for you. It's going to be available. So the key starts 1A, 1B, animal with elongate cylindrical body, pencil-like, body divided into numerous fine segments, or animal body not described as above. So is it elongate and cylindrical, like pencils? No. Oh, well, that would be like a worm, whereas this is not like a worm. So we're going to run through this key. I'm going to show you some neat pictures. Give me a moment, and we'll hop right on that. All right, on to the dichotomous key. So what we have here is a key to the phyla that we'll be going over. So Annelida, Echinodermata, Arthropoda, and Mollusca. If I were you, probably not the worst idea ever to bring up Google and just put in Annelida and look at the annelids, like look at an image search. Bring up Echinodermata, look at an image search. You know, read like the Wikipedia blurb that it'll bring up if you put in Echinodermata. Just give yourself a good background. Annelida, Echinodermata, Arthropoda, and Mollusca. That's going to help you with the rest of this. And then the rest of it's pretty straightforward. So when you run through these, you'll see what I'm talking about. So you can double check your work online. It's very straightforward, like oligochaetes versus Echinoidea, crustacea. It's very straightforward. So again, like here's 8A and 8B. Organisms with 5 or more legs, or organisms with 3 pair of legs. They're very different, in other words. It's very different. So go through the dichotomous key. Use it to fill out this chart. You need to make this chart. And you need to be able to make this chart using this key. So what organisms are you supposed to be looking at to do this? Well, I have built a nice lab PowerPoint for you. And I will be incorporating these images that I've provided into a quiz for you to do online. So if it was me, I would go and pull up my PowerPoint, which is this PowerPoint. It's going to be available to you. Go through, And you can come in here and label all these using your Dichomous Key. So here's the key. You could say it's like mollusca, whatever. All right. Let's see. Yeah, we'll just leave it there. So you can label you can use the Dichomous Key to come in and label all these. And then once you're done labeling all of these using the Dichomous Key, you can go in and do the quiz. That's the way I'd probably do it. It would probably save you some time and decrease your risk of having mistakes. Now, I can guarantee that on your test, I will be providing you with some of these organisms to look at and a Dichomous Key to utilize in order to tell me about the organisms based off of the Dichomous Key. So you'll be, on your exam, there will be several organisms. You have to tell me its phylum, class, and common name. Phylum, class, and common name. And I believe that's the extent of it. So what I want to do real quick is just talk you through the terms that are used in the Dichomous Key, and that should prepare you for going through and doing the rest of it. Alright, vertebrate versus invertebrate should be pretty straightforward. I've provided you with some images here. Things which are vertebrates have a spinal cord with vertebrae, okay, bones in other words, whereas invertebrates do not have an internal skeleton of the fashion I just described. So, for instance, bugs, you know, like fly larvae and spiders and sea stars, they don't have internal skeletons like you have an internal skeleton. Fish and amphibians and turtles, they are vertebrates versus the invertebrates. One of the big deals of this key is the form of symmetry displayed. There is just good old-fashioned being symmetrical. Like here's a spider. You can tell the left side looks like its right side. Your left side and your right side are mirror images of one another, and ergo you are symmetrical. If I were you, I would look up these terms: radial, pentaradial, and bilateral. The idea is that bilateral is two sides, like the spider has bilateral symmetry. Radial symmetry means that something is completely circular and all the, it's got sections, in other words, that all look the same to one another. But one of the big ones that people always screw up is what's called pentaradial symmetry. Okay, pentaradial means it's radial in 5 parts. 1, 2, 3, 4, 5. 1, 2, 3, 4, 5. I've even provided a whole slide on this. You can see the bits. 1, 2, 3, 4, 5. 1, 2, 3, 4, 5. Sea stars. Sea cucumbers, sea urchins, 1, 2, 3, 4, 5. They're all pentaradially symmetrical. All right, they're all pentaradially symmetrical. The idea is that a sea star has legs that stick out that you can see quite clearly as being pentaradial in its symmetry. We call it sea urchin is round but still displays the same basic appearance. A sea cucumber is just a really tall, softer-bodied sea urchin. And if you go and you look up a sand dollar, like sand dollars are the same thing. Sand dollars are the same thing. They all display what's called pentaradial symmetry because they're closely related to one another. Think about your genus, species, all that fun stuff. All right. And let's see what else we have. Elongate or cylindrical, it means worm-like. Like here is a fly larvae. That is going to be an elongate or cylindrical body. Appendages are like arms and legs, okay? Things that can reach out and grab a hold of other things. The spider has appendages. And then the term helical pops up from time to time. Helical means in spiral shape. Like your DNA is a helix, it's in a spiral shape. This is a pretty cool insect. This is a fly. Well, not really a fly. It's a trichopteran larvae. It's a long story. They are called, not lacewings. I forget. Anyway, this is Helicopsyche. Helicopsyche. Caddisflies. These are caddisflies. This is Helicopsyche, and if you look, what it's done is it's made itself a shell that has tiny little stones in it. It's made out of little bitty rocks. There are some really, really pretty caddisflies out there. So that is helical. This is a helical shell, and again, the name of the species is actually Helicopsyche because it makes a shell that is helical in its shape. So be able to use these terms, to apply to these organisms as we go. I tried to provide you with as good images as is physically possible. They're pretty cool. Like here's a leech. You can see it reaching out to grab a hold of things as they do in nature. Pretty neat stuff. Yeah. There's some giant squid images. Everybody loves giant squid. Alright, so go in, knock this out, be able to fill out our chart, phylum class, and common name, and you should be in good shape. Alright, thanks.